Hello everyone, welcome to Molecular Biology as applied to MDLT 499. I'm your instructor, Renan Thompson, and right now we will be speaking about the layout of the Molecular Biology Laboratory, aseptic technique on how to prevent contamination in the Molecular Biology lab. The Molecular Biology Laboratory is normally laid out into three separate and distinct phases, where three distinct activities are undertaken. The pre-amplification area is where cells are normally cultured and DNA extracted and stored. The master mix area is normally where the reagents of the polymerase chain reaction are brought together in one tube, and the amplification area and analysis area is where your DNA is amplified and analyzed. These three areas are very often separated either by time, by personnel, or by geography, or all three. The reason that this is done is to be able to prevent cross-contamination of DNA into different samples. Remember that DNA, in the case of the polymerase chain reaction and its reactions, is the contaminant, but it is ubiquitous throughout the environment. So several steps must be taken to ensure that contaminated DNA does not end up in one's reaction. One of the steps that we take is that many of our steps will occur under a hood. This biological safety hood creates a contained, sterile, and nuclease-free area to ensure that outside and unwanted DNA is not added to the reaction. Before this hood is used, it is very often sterilized with 10% bleach, which degrades DNA, 70% ethanol, which then wipes off the bleach to prevent it from corroding the hood, and then nuclease-free water to wash off any residual bleach and alcohol. All of our implements are regularly wiped off with these solutions as well to ensure that extraneous DNA is not added to your samples. After we are finished working under the hood, we would then turn on the UV light for an hour to be able to degrade any unwanted DNA that may have been left in the hood. Remembering that our extraction and sample processing, our master mixing, and our post-amplification and analysis steps are all done at separate points in the process to prevent DNA cross-contamination. Gloves are also rinsed with 10% bleach and 70% ethanol and changed regularly to prevent this cross-contamination. Pipette tips are always purchased nuclease-free and are always discarded after each use. Pipette tip boxes are only open when necessary and when in use, and they are only exposed to the air when a pipette tip is being exuded from them. Remember that pipette tips are changed after every use.